Hi, everyone. Welcome to today's MCHRI seminar. We're so delighted that you're here. My name is Grant Wells, and here today we will feature Dr. Sarah Bowling, who will be discussing uh, her work related to mapping the routes to building and rebuilding the mammalian heart in health and disease. But first, just want to go through a few quick um, announcements. First, we cannot do this work without our education committee. We're so grateful to them for all the work they put in, volunteering and contributing to these seminars, as well as our symposia and other activities. If you are interested in joining, we would be most welcome if you would consider that. And please consider reaching out to us if that's something you'd like to do. We have about a monthly meeting and then a few odds and ends, different activities, but it's a really great opportunity to get involved. We have another seminar coming up in June. You can register for that on the QR code there featuring Kyle Lowe. Really a wonderful talk that's connected to this that relates to human blood vessels and how they map to vascular development and disease. Really hitting a lot of cardiac activities over the next month with these seminars, which is awesome. Um, we hope you will attend. That'll be on June 16th at noon. And then we'll skip a few months to give everybody a break during the summer. But we'll be back in September featuring this really interesting talk from Jason Cunningham, who is the program director for CERC's Impact Accelerator, which is a really interesting opportunity, I think, just to broaden areas of research that you may have and think about how we transition our work that we do maybe in our clinical studies and translating them into actionable results. So within the community or a health system. So really interesting topic here. That'll be on September 14th. And then an announcement here for the summer scamper. MCHRI has a team. We'd love if you join and consider participating with us. You can register to do so there and contribute to the campaign and get involved and show up and run or go to the community fair. And there's just a lot of really great opportunities to join and participate as a broader maternal and child health community here. We would love it if you would contribute in any capacity that you would desire. Feel free to do so. That's coming up on June 21st. We hope you consider donating to our team as well. And then another really wonderful opportunity as we look towards September, we have our Stanford Medicine Children's Health Research Community event. Another wonderful opportunity to gather as a community. This one specifically, if you've wondered how to collaborate or engage or learn more about what our hospital partners are doing, this is a great opportunity to do that. It'll be Thursday, September 17th from 11:30 to 1:30 p.m. You can register, get involved. It'll be at CAM. We'll have some things to eat and kind of gather and learn more about. We hope you will consider doing so. If there's areas within your research that you've always wanted to see how they might, collaborate with other of our partners within the children's hospital. So please put that on your calendars. We hope you attend. It'll be a great opportunity, kind of over the lunchtime there. And then a really wonderful thing that our marketing team has produced is this MCHRI success story framework. Please consider if you've received funding or publications or different activities resulting from MCHRI's support. Telling us about it. We just want to know. We want to hear all the great work that you're doing. What that helps us do is we can broaden awareness about it and see how our resources and our funding have to your success. Those also help us kind of tell different case studies and stories on our social media. And it's really a wonderful framework to kind of share more about the different impact that MCHRI is having. And then more work that's going on at MCHRI. We have our symposia in October, on October 22nd at Li Ka-shing here on campus. We have our keynote speaker, Dr. Alan Tita from UAB. Our theme this year is correlations as it connects to causation and then how that eventually connects to cures. Some really wonderful speakers from uh, within our own community, but also some invited guests. Registration is open now. You can do so via that QR code. We'll also be announcing that in our newsletter and other emails and communications. So please be on the lookout for this. We hope you will attend. We'd love to see you there. But turning back to why everyone is here today, really pleased to um, host this seminar today. I will pass it to one of um, our education committee members, Dr. Matteo Mole, to do the quick introduction for Sarah. Matteo, over to you.
Thank you, Grant, and I'm very, very excited to introduce Sarah Bowling. She is an assistant professor in the Department of Developmental Biology at Stanford here, and she focused on understanding a mechanism of resilience in mammalian embryogenesis, and in particular, what determines embryo withstand genetic or environmental perturbations. I had the pleasure to meet Sarah many, many years ago back in London when she did her PhD at Imperial College, and she was doing very cool work on the mechanism and roles of cell competition during mammalian development. And then she moved to for her postdoctoral studies to Harvard in the Department of Stem Cell Regenerative Biology and the Boston Children's Hospital, where she developed really state-of-the-art lineage tracing mouse models and techniques for in vivo lineage tracing using cellular barcodes. And she will now give us an overview of what her lab does, combining classical embryological approaches with new next generation tools. And also, finally, also very important to mention that she got very prestigious awards in 2025, the Pew Biomedical Scholar and Baxter scholarship, and also K99 pathway to independence. So very excited to have Sarah here, and we look forward to hear about your research and this seminar.
Awesome. Thanks so much, Matteo. And thanks to the MCHRI for the invitation to speak here today. Can you see the main bit of my slides? Yeah. Okay, great. So broadly, my lab is interested in resilience and mammalian development, and we define resilience as the ability of embryos to withstand and recover from perturbation. So we know that during development, this extraordinary thing happens where a single cell divides and differentiates and eventually gives rise to all the cell types and tissues in the body. But what I think is even more amazing about this process is that it's also incredibly resilient. For example, we know that in really early development, the embryo can split into two, and it actually can, each half of the embryo can go on to give rise to healthy, normal-sized organisms, but two of them. And there's a lot of experimental data indicating that specific organs can tolerate really significant amounts of ablation. For example, there's the study, which I'll talk about more later, in which it's been demonstrated that the developing heart can tolerate up to 60% loss of early cardiac progenitors, but it can recover these missing cells and return to a normal size and function. And then there's a lot of examples in humans as well, where we know from fetal surgeries that fetal wounds can heal scarlessly. And these examples all are in significant contrast with the adult, where wounding often results in scarring, loss of function, or even death. And so in my lab, we're interested in the mechanisms underlying this embryonic resilience. We've known about these examples for a really long time, even hundreds of years when you think about twins, but we still know very little about the mechanisms governing this resilience. For example, we don't understand how missing cells are actually sensed in development. We don't understand how tissues compensate for missing cells. We don't know the extent to which lineages can be plastic during development. We don't know how the tissue sort of knows to stop growing once it's repaired. We don't know what the limits of embryonic resilience are, whether this is something that only happens in certain tissues or at certain windows of development. And ultimately, we don't know why tissues become much less resilient after birth, especially in humans. And so, in understanding these questions, we've sort of hypothesized that maybe there's not just one single route to generating an entire organism from a single cell, but actually potentially many different routes to generating an organism or a cell type from a single cell. And so we're interested in answering this question of how many routes there are to generating a cell or an organism during development. So that if there's roadblocks in the way of one path, then perhaps the organism can take different paths to making tissues. And as a model for these questions, we've focused on the heart, or at least a branch of the lab is focused on the heart. And the reason for this is because, well, there's multiple reasons, but mainly heart development is extremely complex. It involves the migration of multiple pools of progenitor cells that specify quite early in development. So this is a schematic of what the embryo looks like in mouse development around a week after fertilization. And you can see the embryo resembles this sort of cup-shaped structure. It doesn't have much indication of what's going to be the future head or the future tail. So it's just sort of like a mass of cells at this point. But already there's distinct progenitors that are sort of becoming committed towards the heart fates, and they're migrating right across the embryo from the posterior side to the future anterior side. So they migrate across the embryo and then they form this sort of simple cardiac crescent structure that then undergoes this really complex morphogenesis with lots of looping and turning and expansion to eventually form the four-chambered heart. And amazingly, from pretty early in development, this even cardiac crescent structure is functional. So it starts beating really early on. And it has this incredibly important role in pumping blood around the embryo, making sure that the developing tissues are being oxygenated with red blood cells. And because of the way it's formed, it's been described as an aeroplane that's being assembled while it's in flight. So as it's developing, it is expected to function and perform one of the most important functions in the developing embryo. So it's incredibly complex, but amazingly, heart development is also incredibly resilient. So I mentioned this study earlier. This was actually performed in Sean Wu's lab around a decade ago now. And in this paper, they generated these NKX 2.5 CRI lock-stop-locks DTA ES cells. And because NKX 2.5 is expressed in early cardiac progenitors, and diphtheria toxin causes ablation of the cell or cell loss, if you make an embryo out of these cells, then there will be no cardiomyocytes. However, Sean Wu's lab demonstrated that by combining these cells with wild-type cells, you can fully reconstitute the heart, and actually by performing sort of titrations of the amount of wild-type cells that are present with these DTA cells, they found that up to 60% of the cardiomyocytes can be ablated, which is kind of a huge amount, and this doesn't seem to have an effect on the organ function later on. And this has been demonstrated later in development as well, albeit less drastically. But it's been shown, for example, that the early neonatal heart can regenerate following injury that would lead to fibrosis and scarring and cardiac dysfunction later in life. And there's actually evidence this happens in humans as well, where there's been several case reports of early human neonatal hearts having full functional recovery after severe myocardial infarctions. So on the one hand, heart development is incredibly resilient, and there's other examples of this that I'm not even discussing. On the other hand, heart development is incredibly vulnerable in other ways. So we know that congenital heart defects occur in almost 1% of births. There's lots of different types of defects that can occur in humans. And this is an enormous burden on maternal and child health. And so we've been trying to align these two observations. Why, on the one hand, is the heart incredibly resilient to cell loss, while on the other hand, it's very vulnerable. And so to try and make sense of this, we've been really trying to dig into how the heart is made really early in development, as it seems that a lot of these changes that impact heart development later in life can actually arise from really early changes in the embryo. And so, as I mentioned earlier, our current models of heart development describe that there's multiple sources of the heart that specify at the start of gastrulation, just as cells are starting to differentiate in the embryo. And these different lineages of heart cells that give rise to different areas of the heart later in life migrate across the embryo and form this heart. The problem with these models is that we've been really limited by our ability to decipher the origins of the heart and also the plasticity of these different lineages because of the tools that we've been using to understand uh, the fate outcomes of early heart progenitors. So traditionally, these studies have involved using some kind of MCHRI driver, which will enable the labeling of specific populations of progenitor cells, and then the recovery of these embryos to understand which cells in the heart these MCHRI lines are labeling. And it's with a huge number of different MCHRI lines like this, including some which have temporal control through tamoxifen-inducible constructs, that we've been able to put together these models that demonstrate that there are multiple different sources of the heart. However, the problems with these approaches is that they require known markers of progenitor populations. So this essentially becomes a very biased study because you're labeling cell types that you already sort of know about, and so it's very hard to get an unbiased understanding of the different lineages that give rise to the heart. These markers are not always specific, so these can actually lead to really confusing results that actually often disagree with each other from different groups. MCHRIs can also be leaky, meaning that they are labeling cells that have nothing to do with the progenitor population that they should be marking. And they also label progenitors in bulk so that this means that we can't really perform single cell clone labeling. Single cell clone labeling is possible, but it's only possible in a very low throughput fashion where really only one clone or up to a handful of clones can be traced in an embryo at a time. And so all this has meant that we have sort of an incomplete understanding of heart development and normal development, sort of normal unperturbed development, but we also have almost no understanding of how these heart lineages form in any kind of perturbed development, because it's already very difficult to perform this normally. So if you're to translate this into situations where there's some kind of perturbation, such as an ablation or congenital heart defect mutation, it becomes almost impossible. And so because of this, during my postdoc, I worked on generating new tools that enable really high resolution tracing of cell fate choices during development. And so I co-developed models called CARLIN and DARLIN. CARLIN stands for CRISPR-activated repair lineage tracing, and DARLIN is sort of a V2 model of CARLIN. And these models contain what's called DNA barcodes that enable us to label many, many cell clones simultaneously. And in short, these mice contain unedited barcodes that are comprised of 10 different guide RNA target sites. In the presence of Cas9 and guide RNAs that target these sites, double-strand breaks occur in the array, and these get repaired in an error-prone manner to result in a range of insertions and deletions in the barcode. And this combination of indels act as unique and heritable markers of the cell and their progeny. And the real advantages of this approach are that we can generate a huge diversity of information using these barcodes. So we've determined that it's really in the order of billions of unique barcode sequences that can be generated with this model, meaning that in theory, we could label billions of cells at the same time in the same organism. This is also fully inducible barcoding, so we decide exactly when we want to induce barcode labeling. This can be any time point in the embryo or in the adult. All of the transgenic elements are contained within safe harbor loci, which facilitates straightforward crossing of mice to each other and reproducible barcoding dynamics. And all of our barcodes are detectable from both DNA, but also RNA, which means we can detect them from single-cell RNA sequencing assays. And we actually detect barcodes from around 70% of single cells using this approach. And this means that we can barcode embryos, say, mid-gestation. We can then allow these cells to expand so that we generate clones of cells that come from the same progenitor cell. We can then perform single-cell RNA sequencing, and we can determine that the cells that have the same barcode came from the same progenitor cell. And so I want to talk about a project that I started as a postdoc, but has now been spearheaded as by a postdoc in my lab, Marine Secchi. And in this project, we've been mapping fate choices in the whole embryo using cell barcoding. And here we induced barcoding at the start of gastrulation. So just as the heart cells are starting to emerge in the embryo. And then we collected embryos a couple of days later at E9.5, and then we performed single-cell RNA sequencing of different tissues. So we actually selected the head, the trunk, the tail, and the yolk sac through microdissection to perform this single-cell RNA sequencing experiment. And we teamed up with the lab of Kyle Lowe to help annotate these cells. As you can see, there's a huge number of cells here already. There's an enormous amount of cell diversity in the embryo. To make this a bit simpler to read, all the ectodermal cell types, which are going to give rise to the future brain and spinal cord, are labeled in colors of green or shades of green. All the mesodermal cell types, which give rise to the future heart, as well as a lot of internal organs, blood vessels, and other mesenchymal tissues are labeled in shades of red, and all the endodermal cells, which give rise to mostly the gut, are labeled in shades of blue. And so we know which part of the embryo each of these cells comes from because of our microdissection technique. But we can also then overlay our Carlin barcodes on top of here. So here is one Carlin barcode. All of these dots represent a single cell, and all the dots labeled purple here come from the same progenitor cell because they all share the same barcode. And so in this Carlin clone, it's comprised mainly of cells within the brain tissues as well as the neural crest. So it's labeled some kind of early ectodermal progenitor. In this other clone, we see this largely restricted to the mesoderm. And here we have another clone which is restricted more to the extraembryonic mesoderm, which is going to make the really early blood in the embryo. And so when we first got these data, it was really exciting because we were suddenly able to see hundreds to even thousands of clonal behaviors in one embryo. But one of the issues was actually in its complexity. It became very hard to analyze by just looking through these barcodes one by one. And so I actually collaborated with Deborah Van Eggeren, who is a postdoc, is now a postdoc at Stanford and worked in as a PhD student in my postdoc lab. And she came up with the idea to actually cluster these barcodes into distinct behaviors. And this is using an approach called principal component analysis. And here is one of these plots where each barcode is represented by a single dot here. And then barcodes are sort of spread out in space based on which cell types they appear in the data set. We can then cluster these barcodes and put these barcodes back onto the UMAP. And we can use this to actually visualize these clonal restrictions that occur in early development. And I'm just going to point out a few interesting examples here. So one is these neuromesodermal progenitor clones. These are cells that give rise to both spinal cord, so an ectodermal cell type, as well as the Semitic mesoderm, which is a mesodermal cell type. So these have been described a few years ago, and they were really exciting at the time because they sort of defied our expectations of uh, uh, lineage commitment during gastrulation, where we expect cells to differentiate into either mesoderm, ectoderm, or endoderm. But these cell types sort of defy these expectations and give rise to two different germ layers for quite a long period during gastrulation. We also see lineages of the neural crest that give rise to really diverse cell types in the embryo. You can see they're on opposite sides of the UMAP. And we can also see clonal couplings between the yolk sac, the early blood, and the endothelial cells in the yolk sac as well. So we've decided to use this to focus more on mesodermal populations during gastrulation. And one of the really striking things about the data set was that it really demonstrated this massive divergence of mesodermal populations at this stage. So this is one of the ways in which we present these data commonly. It's a heat map, and each row represents a unique barcode, and each column represents a unique cell type. And I'm not actually labeling the columns because it would make the plot too small and confusing. So I'm just going to take you through different cell types or groups of clones at a time. And one of the things I want you to take away from this is that if we have a clone that gives rise to all cells in our data set, then we will see one big line across the heat map. But hopefully what you can appreciate is that we don't see that very much at all. In fact, we seem to get different groups of clones that give rise to completely different cell types. So, for example, we get this group of clones that give rise to purely extraembryonic mesoderm. We get a distinct group of cells that gives rise to red blood cells, another group of cells that give rise to hind limb, to cranial mesoderm, to cardiac mesoderm, to the forelimb, to Semitic mesoderm, and paraxial mesoderm. So to put this into context, the embryo looks like this at the time that we're labeling. So it's very small. It's around 200 microns in length. And already it's established, or there's sort of fate established in this embryo that is going to give rise to all these different cell types. And this actually really nicely echoed some really early work that was performed on mouse embryos by injecting dyes into different regions of the embryo. And this early work demonstrated that different regions of the embryo are sort of fated to become different cell types. So, for example, in this region of the embryo, the really posterior part, generally these cells become extraembryonic mesoderm, while slightly lower down, these cells generally become lateral plate mesoderm, and so on. So it was really reassuring to see these clonal interactions that we expected from the previous literature, but it was also kind of eye-opening to realize how much diversity of clones emerges really early on in the embryo. So we want to talk about two examples in which we've focused on specific cell types, and one is the endothelial cells. So the endothelial cells are cells which line the blood vessels of the embryo. And they're found everywhere in the embryo. So, of course, blood vessels are critical for transporting blood around the body to ensure that the developing tissues become oxygenated. If we perform this labeling with CD31, an endothelial cell marker, we can see that these are pretty much all over the embryo. And so a long question has been that of where endothelial cells actually come from in the embryo. It is thought, in some schools of thought, that they come from one origin, and then these cells sort of migrate around the embryo. But it was less clear the extent to which they can also emerge locally in the embryo. And so we were able to identify endothelial cells very clearly in our data set. They are marked by this gene CDH5, which encodes for cadherin. And when we look at our clonal clustering, we can see that actually they seem to come from multiple different progenitors because they belong to multiple different clonal clusters rather than just a single clonal cluster. And to some extent, this seemed to overlap with the tissue of origin. And so we were interested to see whether these endothelial cells were actually generated from local progenitors. So to test this, we we generated a heat map again here. So this is just looking at endothelial cells in different areas of the embryo. And you can see there's actually not very much overlap in the clones of the endothelial cells from different areas of the embryo, suggesting that they do not come from a common origin. And in fact, when we looked at non-endothelial cells from the same areas of the embryo, this supported this idea that endothelial cells are generated from local progenitors. So the head endothelial cells generally have most clonal overlap with head non-endothelial cells, and trunk endothelial cells generally have most clonal overlap with trunk non-endothelial cells, and so on. And to ask this question more quantitatively, we performed this lineage coupling score, which confirmed that endothelial cells from each part of the embryo had far more clonal overlap with non-endothelial cells from the same part of the embryo than with endothelial cells from different parts of the embryo. We went a bit further to see whether they actually are associated with different transcriptional profiles. And so for this, we subclustered all the endothelial cells in the data set, and we performed differential gene expression analysis. And this demonstrated that already at this really early stage in development, there are distinct transcriptional signatures that are associated with distinct endothelial cells in distinct areas of the embryo. We were also able to use our clonal profiling to actually determine that there was even a higher number of sort of different types of endothelial cells within different areas of the embryo. And we could see that these were also associated with distinct transcriptional profiles. For example, endothelial cells associated with the yolk sac or extraembryonic tissues had this increased expression of FOXF1, a transcription factor, while endothelial cells associated with the hind limb, for example, had higher levels of this gene ifetin1, which is associated with the posterior areas of the body. So to conclude here, we've been able to demonstrate that endothelial cells are generated from local progenitors throughout the embryo that give rise to distinct non-endothelial cell types. And we've also found that endothelial cell transcriptional state is influenced by site and or cellular origin. And I think this has really important consequences for generating endothelial cells in vitro. This is actually a big area of research, and I think we'll learn more about this when Kyle speaks in a couple of weeks. But in order to make blood cells come from endothelial cells, and so we need to sort of optimize our protocols for endothelial cell development in vitro. And this data suggests that actually there's not one common endothelial cell state, but actually you get a different kind of endothelial cell depending on which area of the embryo that you're in. And so I think this is really important to bear in mind when we perform these experiments of optimizing in vitro endothelial cell production. This has also been reassuring because recent studies have used sort of lineage inference methods to infer the origin of endothelial cells. And this is done by collecting embryos at really close developmental time points and determining at what point they seem to diversify. And recent studies have been unable to determine that endothelial cells actually come from different sources through this kind of approach. So I think it's really demonstrated the importance of these ground truth clonal tracing experiments in determining the actual origins of cell types in the embryo. So moving on, I want to talk a little bit about how we've used the same data set to determine the clonal origins of the heart. So as I mentioned earlier, there's multiple different origins of the heart, and we wanted to determine whether we could distinguish these distinct origins for the heart in our barcoding data. So for this, we collected embryos at the same time. We also added a specific library that encapsulated just cells of the heart so that we could really enrich for these cardiomyocytes. and we performed single-cell RNA sequencing. And first, we looked at the clonal cousins of the cardiomyocytes in our data set. So here's a heat map where we have all the cardiomyocytes. We see two different populations of cardiomyocytes that were a little bit transcriptionally different. We just labeled them here 1 and 2. And then we looked at what their clonal cousins were, so which other cell types were in each clone that gave rise to the cardiomyocytes. And we found that there was a pattern here where there was a small population of cardiomyocytes that had cousins in the extraembryonic mesoderm, so the yolk sac and the amniotic mesoderm in early blood. There was then a distinct group of blood cells that had cousins that were in the embryo that gave rise to the pharyngeal mesoderm and lateral mesoderm. And then there was a group of cardiomyocytes that had no sharing whatsoever with other cell types in the data set. And we were intrigued about this because some really recent work from the last few years has actually demonstrated that there could be a progenitor in the embryo that has dual capacity to give rise to the yolk sac and to cardiomyocytes. And this was quite controversial when it came out. And so it was kind of intriguing that we were able to see the exact same thing when we performed this very unbiased lineage tracing. And the authors of this study demonstrated that this yolk sac linked cardiomyocyte mainly gave rise to cells of the left ventricle and atrioventricular tract, whereas the later emerging progenitors that they think is more consistent with the second heart field are more biased to giving rise to the right ventricle and the atria. So we took our cardiomyocytes and we annotated them according to whether they look like ventricular cardiomyocytes or outflow tract cardiomyocytes or atrioventricular canal cardiomyocytes. And then we overlaid our different clonal identities on top of here. So these are clones that were found in cardiomyocytes that were linked to the extraembryonic mesoderm. And we found that these were really biased towards specific cardiomyocytes in the data set, so largely cardiomyocytes in the ventricle and in the atrioventricular canal. Meanwhile, the embryonic mesoderm linked clones were much more enriched in the outflow tract and the atria. So this actually really nicely recapitulated the work of this published study, suggesting that these do have different profiles. And when we looked at the no-sharing clones, this had a pattern of overlap that was much more similar to the extraembryonic mesoderm link clones. We can show this, that there's sort of this lack of overlap between these different clonal groups when we look at them all together on the same UMAP. And so together this suggests that in the embryo at E7.5, there's already progenitors that have sort of diverged in fate that give rise to either the extraembryonic mesoderm and to the cardiomyocytes, or to the progenitors for embryonic mesoderm and cardiomyocytes, or these progenitors that don't have any sharing with other cells in the embryo that could be restricted progenitors for cardiac fate. And again, the pattern of overlap we saw seems to recapitulate the previously described diversification of fates at this stage. So there's a lot of non-cardiac cells in the heart as well, and so we're intrigued as to whether these lineages could retain the potential to give rise to non-cardiac cells at the heart. And so for this, we additionally clustered the non-cardiac cells that can be found in the heart, including the epicardium, the layer of cells around the heart, as well as the endocardium, endothelium. And we looked at these different clonal groupings. And we found that actually these clones were all associated with both endothelial cells and with epicardial cells. So what this demonstrates is that actually, even after the point at which fates have diverged to give rise to different regions of the heart, they still retain the capacity to give rise to non-cardiac cells, including the endothelium and the epicardium. And this also demonstrates that there's multiple sources of both endothelial cells and epicardial cells in the embryo. So to summarize this part, data has supported a divergence in cardiomyocyte progenitor fates by E7.5 in the embryo that give rise to distinct domains of the heart. And these cardiac progenitors have diverse fate outcomes in the embryo, from the yolk sac mesoderm to pharyngeal mesoderm. And after divergence, our data suggests that progenitors retain the capacity to give rise to non-cardiac cells in the heart, including the epicardium and endothelial cells. And so I won't show all the examples, but actually in analyzing these data, we found multiple examples of this kind of lineage convergence during embryo development. So this was in the endothelium and the cardiomyocytes and epicardium that I've showed already, but also in the Semitic mesoderm and in the allantois. So what this suggests is that many organs in the embryo are actually comprised of multiple different lineages. And going back to the bigger kind of question in the lab, we were intrigued as to whether this kind of lineage convergence could act as a mechanism of organismal resilience by providing cell type redundancy. So, for instance, if we lose one of these lineages through mutation or through some kind of damage to the embryo, is it possible that then the other lineages can sort of step up and increase their output to actually still continue to form a functional organ? So this brings me full circle to the original question of understanding how our tissues withstand and recover from perturbation, and to establish these different routes to generating a cell or an organism. So in the last few minutes, I want to talk about how we started to investigate that. So firstly, we're interested in how the heart compensates for missing cells during development. And this is really related to that study that I showed at the beginning from Sean Wu's lab, where we know that the heart can withstand loss of up to 60% of cells during development. And so this project is being led by a graduate student in the lab, Peter Martin. And the goal of this project is to perform the same kind of cardiomyocyte barcoding, but in the context of some kind of ablation that removes a lot of cells in the embryo, to see which cells are actually responsible for coordinating the recovery of the embryo. And actually, the hardest thing about this project has been developing ways to ablate cells in a really defined fashion. And so Peter has been exploring different approaches. One is through using MCHRI-based ablation of cells using diphtheria toxin, which, as soon as expressed in the cell, should cause the cell to die. And looking at the literature, there was this paper published around 10 years ago in which the authors generated this MCHRI line that is under the control of a hand-one enhancer. And this leads to fairly restricted expression of the transgene in the left ventricle. And the authors demonstrated in just a small panel of the paper that if they crossed this MCHRI to a diphtheria toxin allele, meaning that all the cells here that are blue should die, then they found that the left ventricle was slightly smaller at E12.5, but then at E16.5, the left ventricle seemed to be a normal shape and size. And so we thought this could be a really good contender for answering some of the questions that we have about plasticity of lineages in the embryo, especially given that the yolk sac linked progenitor population gives rise largely to the left ventricle. And so Peter imported this mouse, and you can see here he crossed it with a TD tomato reporter line, and he was able to recapitulate this really specific staining in the left ventricle. He then performed a time course of development where he either looked at the TD tomato expression or crossed these cells with a diphtheria toxin allele, where all the cells that are in red should be ablated, and these images at the bottom. And you can see that it does seem like there might be some small changes to the left ventricle size. So at E10.5, it looks a bit smaller. It seems to have a hyperplastic right ventricle at some points as well, at E11 and E12. But by the end of this time course at E14, he wasn't able to see any differences in the size of the left ventricle. So we still have questions as to the specificity of this allele as well as the efficiency of the diphtheria toxin production. But these experiments are ongoing where he's performing this kind of ablation in a barcoded mouse as well. His other approach has been to perform drug-induced cell death, and this actually was inspired by studies performed almost 50 years ago now, in which the early mouse embryo at the start of gastrulation was treated with this drug mitomycin C to cause a huge amount of cell loss in the embryo. And if we look at cell number in different lineages of the embryo, you can see there's this massive reduction in cells. And amazingly, the authors found that there was also this increase in mitotic activity, suggesting that these cells are actually increasing their proliferation following cell death. They expected the embryos to be either not alive or to be very defective. But actually, this original study found that although the mitomycin C treated embryos are smaller and delayed for some time, they actually end up catching up and recovering to a similar size to normal unperturbed embryos. So Peter has been repeating these experiments, and he has been able to recapitulate these phenotypes of this delayed embryo growth. And amazingly, he's also been able to find that by a later stage in development, the embryos seem to recover to a similar size, or at least weight, and morphology to the PBS control treated embryos. So I think this is also another potential exciting model in which Peter can study how these lineages reform after this massive cell loss. And the goal here would be to induce the same barcoding at the time point that we've already carried out, but with some kind of cell ablation to see how these lineage maps change. Finally, we are also interested in vulnerability in development. And so as part of this, we've been exploring factors or mutations that are associated with heart defects in humans. And actually, one of the environmental impacts that has come up that seems to be highly associated with heart defects is maternal hyperglycemia. So this is actually incredibly common in pregnancy. It's thought that up to 15% of live births are affected by some kind of hyperglycemia during pregnancy. That includes hyperglycemia from type 1 diabetes, from type 2 diabetes, or from gestational diabetes. So this is very common, and it is becoming increasingly common as well as diabetes rates are rising in the population. It's associated with an increase in birth defects in humans, including neural tube defects, craniofacial defects, and cardiac defects. This is a plot showing this massive increase in the rate of cardiac birth defects as you increase the glucose concentrations in the mother. And it's unclear the mechanism of how hyperglycemia impacts development. And part of this is because it's been very hard to model this in mice with the same kind of control that we have, or the same kind of nuances that we see in humans. Because our current models of hyperglycemia or diabetes in mice involve either the use of drugs that cause ablation of beta cells, or the use of genetic models that have reduced levels of insulin, or the use of diets that just result in obesity and hyperglycemia. However, the problems with these approaches is that the perturbations are associated themselves with reduced fertility. So it's actually very technically challenging to get these mice pregnant, which makes these studies very slow. There's a lot of off-target effects associated with all of these perturbations as well. The penetrance is not always complete. And most importantly, there's no ability to model hyperglycemia at specific windows of development. So basically, the mice are hyperglycemic from the moment that they become pregnant. And there's very limited opportunity to sort of fine-tune the window of hyperglycemia. And so a postdoc in my lab is Kowalczyk, who actually recently is a MCHRI postdoctoral fellowship awardee, has been developing a new way to generate or study hyperglycemia during embryogenesis. And this is a collaboration with Sung Kim, who's also in the Department of Developmental Biology. And she's been using this Black 6 RIP-DTR model, in which beta cells express the diphtheria toxin receptor. And when diphtheria toxin is added to the mouse, then the beta cells are ablated, and this results in quite swift increase in blood glucose levels. And so EASE has been able to demonstrate that within just a couple of days of inducing the mice with diphtheria toxin, there's this massive increase in blood glucose levels. Anything over 200 is thought to be hyperglycemic. And so these mice are exposed pretty rapidly and with very high penetrance to hyperglycemia. And you can see that there's this really nice temporal control where she can decide exactly when these mice are exposed to hyperglycemia. She's been able to track the phenotypes associated with hyperglycemia exposure at different time points in development. She's been able to see that really early onset of hyperglycemia is associated with massively reduced embryo size and weight, as well as quite severe neural tube defects, while these defects become less significant with later timing of induction. And finally, she's been studying the impact on heart development. Again, she sees that the early induction of maternal diabetes results in really deformed hearts that have septal defects as well as overgrown cushions that haven't formed proper valves. Whereas these embryos look a lot more normal at later time points. And she's currently investigating the types of defects that emerge from this later hyperglycemia onset. I'll skip this last slide. So overall, I hope I've given you a good overview of what we're interested in the lab. From the lineage tracing point of view, we're interested in generating these really high resolution, unbiased maps of lineage origins of the mammalian heart. We want to use this to determine the lineage changes in the context of hyperglycemia and in mouse backgrounds that have increased rates of congenital heart defects. We're also interested in these mechanisms of resilience and vulnerability, including understanding how missing cells and lineages are recovered in development following ablation, understanding mechanisms of regeneration in the heart, and investigating these questions about the new routes to generating cells in vitro, and hopefully gaining a better understanding of the variable penetrance of congenital heart defects and variability in normal heart development as well. So with that, I want to thank everyone in the lab. I want to thank my collaborators, my funders, including the MCHRI, who generously awarded us a pilot award for this work. And thank you to everyone for listening.
Thank you so much, Sarah. That's wonderful. I'll open to questions. If anyone would like to ask a question, maybe they can unmute and ask directly, or if they prefer, they could just write in the chat, and I'll do my best to answer. Not answer. Answer will be Sarah. Let me just check if there is anything in the chat. No, I don't see.
Hi, Sarah. I have a question.
Hi.
I was wondering for I think I find it very interesting to cluster the Carlin lineage cells by their barcodes. Can you explain a bit more about this? I presume this is at the sequence level at the single loci.
It's actually, I think it's a bit more simple than that. So we just take a matrix of the barcodes and which cells they appear in, and then that is the information used to drive the principal component analysis. So if you have a bunch of barcodes that always appear in a cardiomyocyte and a yolk sac cell, then they are going to cluster together. Whereas if you get another bunch of barcodes that always appear in the forebrain, then they'll cluster separately. Our approach is pretty basic. There are new tools that have actually uh, just been put into a preprint. It's called Clone2Vec, in case you're in that field, but I think it's a more mathematically robust way of doing the same thing. So if you're interested in that, then I can share that, or you can look that up.
Yeah, that would be interesting. We are currently using the Darlin system, and was wondering whether it would be applicable to use this method for identifying further analyzing lineage relationships. I presume then that, say, if two clones have about 98% similarity, you would then sort of cluster them more similarly to each other.
Exactly, yeah. Yeah. And then it becomes the cluster resolution, like how much you want
So,
to break up the clones
okay,
based on
awesome.
the similarity. Yeah.
Oh, thank you so much.
I spoke for quite a long time, so.
Any other question?
Hi, Sarah. This is Lee. It's fantastic talk. I have a quick question regarding kind of the time window you usually use to induce the sparkle. Like how long? I see in your examples you have tried maybe 3 to 4 days. Um, right. What's the concern about that?
Yeah, so we actually just do a single induction of barcoding with a single dose of doxycycline, which triggers the Cas9 and the barcodes to emerge. We think this barcoding takes place over the course of around 24 hours. And actually, by 12 hours, 60 to 80% of the barcodes are already labeled. So the dynamics are fairly quick in development as well. We could, if we wanted, perform multiple induction injections, so give doxycycline at multiple time points, and then this would allow us to build up more complex phylogenetic trees. The reason we haven't done that is because I think then it becomes very technically challenging to actually reconstruct these trees. Probably you're familiar with it a bit, but it becomes basically a probability based metric of what trees are more likely versus less likely. Whereas we really like the fact that we have absolute confidence in two cells that are clonally related to each other.
Oh, gotcha. Thank you.
If there are no more questions, probably we end here considering the timing. Thank you so much, Sarah, for the wonderful seminar, and wish you a great day to everyone. Thank you.
Thank you.
Bye bye.
Everyone, thank you.
Bye.